Hello, everyone. My name is Mark Fischetti. I'm a senior editor at Scientific American magazine. Thank you for joining us to learn more about the coronavirus. Our expert guide today is Britt Glaunsinger, who is a virologist at the University of California, Berkeley and Howard Hughes Medical Institute. And she's a specialist in infectious viruses. I'll have Britt introduce herself in a moment. First, a couple of quick housekeeping notes. If at any time you're having technical difficulties, please contact us through the chat window. And if you have questions you'd like us to consider, put them in the questions window. The broadcast is an hour and a half, probably about halfway through. We'll move to Q&A with you all. So, but feel free to enter those at any time. So Britt, let us know a little bit about yourself and then we'll get started.
Yeah, thanks, Mark, and thanks for having me today onto the panel. As you indicated, I'm a virologist. I've actually studied viruses for about the last 25 years, which is pretty much my entire career. I became captivated by them when I was an undergraduate student at the University of Arizona. I had read Richard Preston's book, The Hot Zone, which maybe some of our audience members have read too. It's about the early Ebola virus outbreaks. And that was sort of my first exposure to thinking about viruses and what really fascinating entities they are, given they have, compared to our genomes, really small sets of genomes that can do a remarkable amount when they come in contact with humans or other hosts. And so I then went on and got a PhD in virology at Baylor College of Medicine, and then did postdoctoral research in virology at the University of California, San Francisco, before starting up my own research group as a professor at the University of California, Berkeley. And my lab actually most of the time studies herpes viruses from a research aspect, although recently we've been part of a large coordinated effort on the Berkeley campus that's been organized by our Innovative Genomics Institute that's headed by Jennifer Doudna to understand holistically COVID-19 and this new coronavirus. And so we're part of a group of labs who are studying the basic biology of how the virus works. That's really in my wheelhouse, as well as trying to develop antiviral therapies to stop the virus. They've got a lab set up to do community testing, and there are several members of my group that volunteer there, as well as doing serological testing and understanding in the community. So it's really sort of a comprehensive effort that I've been part of on the coronavirus front there. And I also teach about coronaviruses and many other viruses that I've done for about 15 years and have published a couple of papers on them, including this one with Scientific American that came out, I think, in the July issue. And there's a wonderful interactive web-based version that we're going to be covering in the webinar today. And so I hope that I can demystify some aspects of how this virus works and how scientists are trying to stop it, and answer a bunch of questions.
Great. Yeah, that's great. Yeah, I want to say, so everyone listening knows, Britt was a tremendous help to us. The interactive, which you'll see behind us at certain points, it just really digs into how the virus works. And we're going to have Britt work through that. I want to thank a few people who also worked on all of this together. The freelance artist, Veronica Falconieri-Hayes, was huge in making these models that you'll see. Scientific American's graphics editor, Jen Christensen, and our multimedia editor, Jeff Delicio. Britt's going to break down the graphics step by step, explain how the coronavirus sneaks into our body, how our immune system works, how the virus can sometimes defeat our immune system, and how the leading drug candidates and vaccine candidates seem to be working, what their approaches are, and then we'll take your questions. I also want to thank Sonia Buda, who's behind the scenes here, our engagement editor, who's running the various windows, and Macarena Carrizosa, who's running the slides and videos. So to get started, we're going to begin with you, our viewers. We're going to take an audience poll. We're going to do 3 of these during the webinar. We'll pose a question, then a window will appear with choices. You have about 30 seconds, and then we'll all see the outcome together. Please know all the responses are completely anonymous. So Macarena, could you please start the first poll? while you're answering, we'll all just ponder. I guess someone's going to let us know about when we're ready for answers, right?
Luckily, no one's getting graded on this.
Right. Right.
It's a low stress class today.
There you go. Right. Okay, so we're back. Interesting. So I think we all can see this, but just to sum up, in case it's hard to read if you're on a phone or something, where can the coronavirus reside in your body? Multiple answers were okay. So 42% said nose, throat, and lungs. 8% said stomach, intestines. 9% said heart and other organs, and 65% said all of the above. So, Britt, tell us, even though we're not being graded, tell us the answer.
The answer is probably all of the above. I mean, we hear most about the respiratory aspect of the infection. We know the virus replicates in the nose, throat, and lungs, but there is clearly some evidence that it can spread beyond those regions to other parts of the body as well. It is important to distinguish, though, that you may have symptoms in areas of your body that are a reflection of an infection elsewhere. So you may have heard in the news about things that are, dementia related or brain related. And I think the evidence is not yet clear as to whether the virus can actually replicate there, or that's a sign, sort of a downstream consequence of an infection somewhere else. But I think emerging evidence is pretty indicative that the virus is not just restricted to the respiratory tract.
Great. Okay, good. Well, so we'll do 2 more of these, everyone. The next one's going to be a bit of a surprise question too. So let's start. And Britt's going to talk to each of these slides, and we'll have Macarena change them when we're ready to go. So Britt, I'm going to turn this over to you right now.
Yeah, so you can go ahead and switch slides. That first one is just kind of giving you a bit of a primer for what that interactive looks like on the web. What I wanted to start with was just kind of to introduce you to the coronavirus and viruses in general. So if you could switch to the next slide, please. We'll start with the question of what is a virus? So next slide. And I want to think about, viruses as things that, oops, sorry, one too many, the prior one. Viruses have a lot of things in common with living organisms, but we actually don't consider them to actually be alive. Viruses are inert unless they can find a cell to get into. So all viruses have a couple of things in common, and that is that they have to have a set of instructions, a set of genetic instructions. In our own cells, these are DNA instructions in our genomes, and some viruses have DNA genomes. But other viruses have their set of instructions in a molecule called RNA, and this is the case for coronavirus. And so if you don't know what RNA is a nucleic acid like DNA, but in our own cells, those DNA instructions have to get copied or transcribed into RNA, because that is the message that can, or the language that can be read by the cell's protein making machine, which is called the ribosome. So our own messages go through an RNA intermediate, and some viruses just start out with their set of genetic instructions as RNA. So every virus has a set of instructions. That set of instructions is generally 1 million times or so smaller than the set of instructions in the cells that they infect. So remarkably compact set of instructions that can really pack a punch. And then every virus has to protect that set of instructions with a coat. You can think of this as the house for those instructions. And so that can either be a coat that's made just out of proteins, or a coat that's sort of a mixture of lipids, which are fats, and proteins, and this would be called an envelope, and that is the type of coat that the coronavirus has. So if you look at these slice away pictures, which are just sort of taking an image of a snapshot of what the inside of the coronavirus looks like, what we've done here is just highlighted the different components of the actual virus particle that's floating around before it finds a cell to infect. So you'll see that the RNA genome is highlighted in orange on the upper right-hand side here. This is the genetic instructions. It's sort of a fun fact about coronaviruses. They have a remarkably large genome for an RNA virus. In fact, it's about twice the size of your average RNA virus. And one thing that means is that the coronavirus can encode more genes, so make more viral proteins that can do more things than many other RNA viruses can do. And they can manipulate the cell in different ways than you might expect other RNA viruses to do, because they have an unusually large genome for an RNA virus. And then that genome is coated with a protein called the N, or nucleocapsid protein, that's shown in blue in the center upper right. This is something that's just sort of protecting the genome itself, helping to make sure when new viral particles are formed, helping to make sure that that genome gets encapsulated into that protective coat. And then this is surrounded by a lipid or a fat envelope that's stolen from the host cell. No virus can make their own lipids or encodes for things that can make lipids. So anytime you see lipids in a virus, those are taken from the host, and that's certainly the case for the coronavirus. So the envelope, which is shown in the center lower panel in sort of a peach color, and then studded into the envelope are a number of different proteins that are viral proteins. There's the matrix or M protein shown in purple. This is something that helps with formation of that virus particle in cells, as does the E protein, which is shown in yellow on the lower right-hand side. But the one that we're going to talk a lot about today, and that I'm sure everyone has heard about, is the famous spike protein, this long orange in this diagram protein that's sticking out. And the spike protein is actually a feature of coronaviruses, which gave them their name. So viruses can only be seen under an electron microscope. They're too small to see under a light microscope. So if you look at a coronavirus under an electron microscope, you researchers would see this sort of notable structure that was due to these spike proteins sticking out that made the virus particle look like a crown or a halo, a corona. And so that is how coronaviruses actually got their name. But beyond being sort of physically obvious components of the virus, the spike protein does two really important things for the virus, which is why there's been so much research attention focused on the spike protein as an avenue for inhibiting the replication and entry by this virus. So let's move on to the next slide and talk a little bit more about what it is the spike protein is doing. So the core job of the spike protein is to get that virus particle, which is inert, unable to do anything on its own, get it into a cell to help the virus invade a cell, because within the cell, the virus almost comes alive. And it will then be able to transform that cell into a virus-making factory. But it can't do anything until it finds a cell to get into, and then somehow finds its way past that barrier that's the surface of the cell to get inside. And so the spike protein is crucial for both of those activities. So what you'll notice in this image, and in just a minute, I'm going to show a kind of a video of this, but let me describe what you're going to see first. So let's take a look at the spike protein. You'll notice it looks kind of like a stalk, right? And at the tip of that stalk, the head is sort of this globular domain. And what that is doing is you can think of as like a key. And that key is looking for just the right lock to fit into. because just the right lock means it's found a cell that it can somehow gain access to because it's got the right key. And the lock is this ACE2 receptor that you've probably heard of. So what the spike protein needs to do first is locate cells that have this right lock, this ACE2 receptor, so that it can dock onto them and unlock the cell. So that binding, that inserting into the lock is the first job of the spike protein. Without it, the virus would never be able to even locate a cell where it could get in. But that's not all it does. So just inserting the key into the lock, well, that tells you you've got to the right place, but it doesn't open the door to get you inside. And so there's a second hidden activity within this spike protein, which is actually a group of 3 proteins together. It's called a trimer. And what that is a machine, like a spring-loaded machine that allows the virus inside. It's called a fusion machine, and it's called a fusion machine because what it does is it allows the virus envelope membrane to fuse with the cellular membrane. So let's take a look at what happens here. Let's move the slide so we can see this first animation. All right, so what's happening here is you'll see that the tip of the spike protein binds the ACE2 receptor. But then in order to turn on that fusion machine, a second thing has to happen, which is that the tip of that spike protein gets chopped off. And it gets chopped off by a set of molecular scissors that is called a protease. And that's what turns on this fusion machine, allows it to unfold, insert into the cell's membrane, and bring those 2 membranes together so that the virus can now deliver its genome payload into the cytoplasm of cells. So spike is doing 2 really critical things. It's finding the cell and it's opening that door through its spring-loaded fusion peptides that are sort of buried in the center of that spike complex. So that's why so much effort has been focused on that, because if you can stop either that binding event or the subsequent fusion event, then the virus is, for all practical purposes, inactivated. It can't do anything if it can't get into the cell. It can't cause disease, it can't replicate. So that's a really important target. All right, let's move on to the next slide and talk about what happens once that genome payload is actually delivered into the cytoplasm of the cell. Now's when all the action is happening on the virus front. You're moving from an inert particle that's doing very little on its own, it's inactive, to as soon as it gets into the inside of the cell, there's a flurry of activity that is happening in really short order. The virus basically takes its genome, which is this RNA genome, and recruits the cell's protein-making machine, which is called the ribosome, to turn that viral genome into viral proteins. Proteins are the molecules in a cell that are basically the workhorses. They're the things that are getting things done. So by turning the genome into proteins, now the virus is able to start using those proteins to transform the cell into basically a virus-making factory. And it does this in a few ways. One is that it starts to remodel the inside of the cell. It actually creates its own rooms, these little vesicles that you can see here drawn in light blue that look like circles. It's creating its own rooms where that RNA, the virus genome, can now be copied lots of times. And it's copied because one of those, the first sets of viral proteins that are made are proteins that every RNA virus has to bring in or encode. And these are proteins that form the RNA copying machine, which is called an RNA-dependent RNA polymerase, or just a polymerase. The polymerase is a core feature of viruses and RNA viruses, and it uses this. You can just think of it as a molecular copying machine. It will take one copy of RNA and make hundreds and thousands of copies. And those copies can then be used to make lots more viral proteins, which are doing the work of hiding and blocking the immune system, are doing the work of copying more viral RNA, are doing the work of forming new viral particles. You're making lots of spike proteins, you're making lots of nucleocapsid proteins that are all components of the viral life cycle. So if we move along in this animation to the next part, you can slide down and see that, okay, so we've got lots of copies of the RNA, and what the virus needs to do now is basically package those, form that protective house around them so that they can get out of the cell and be released. And so they're doing this through another series of vesicles that are in the cell that are basically studded with the spike protein that is being made inside the cell. And the virus is coming in and through interactions between the viral genome and the nucleocapsid protein and proteins that are part of that outer envelope, it's basically getting enveloped and forming these little packages of lots of new virus particles, which are then released from the cell, where they can go on to either infect neighboring cells or be breathed out or coughed out into the environment to get into another person and start a new infection there. So that's the basics of what happens once the viral RNA gets into the cytoplasm of cells. You can start with one infectious particle and get hundreds or thousands of new virus particles that get released from that.
Hey, Britt, I have a quick question. So we're going to go now to how the immune system responds to all this. There's a lot of talk about mutations, and what you just described seems pretty neat, I almost want to say. But can the coronavirus change over time and alter how this whole process works, which makes it then more difficult for the body to recognize that? I don't want to get too far.
Yeah, so mutations are a really central feature of pretty much all viruses, and RNA viruses in particular mutate usually very, very fast. And this is why we have so few antiviral drugs, is because it's really difficult to engineer drugs to viruses because they can change so quickly. So if we think about viruses like HIV, which has an astronomically high mutation rate, or influenza, very high mutation rate, it means that the virus is continually changing over time. That's one of the reasons why we have to get a new flu vaccine each year, is because the virus changes enough that our immune system, doesn't recognize it as exactly the same and has to build up new immunity each year. So the reason why RNA viruses mutate so quickly has to do with that polymerase, the RNA copying machine. For almost all RNA viruses that we know of, hundreds and hundreds of them, that RNA copying machine makes errors. It's not perfect. In our cells, our DNA copying machine makes errors too, but it's got built-in correction mechanisms, proofreading mechanisms, which means that if it makes an error, it's got some whiteout that it can go and correct it. Just hit the delete button and put the right thing in. But most RNA viruses can't correct those errors. So just naturally, as the copying machine is going along, randomly, it's making mistakes. These are not directed mistakes, they're random mistakes. And sometimes those random mistakes are bad news for the virus. It makes for a virus that is worse off. And so those will just sort of fall out of the population. Very rarely, but occasionally, those mistakes tend to give some advantage to the virus under a particular situation, allows it to evade the immune system in a new way or something like that. And these are the ones that then will propagate. Turns out that coronaviruses make fewer mistakes than other types of RNA viruses because they unusually have a special correction, proofreading activity to allow them to correct some of those mistakes. So we look at sequences, and in fact, now there's something like 30,000 different, or maybe more than that, many tens of thousands of viral sequences that have been analyzed by scientists from individuals who are infected. And so what they do is they sequence the virus and they look to see how many changes are happening, how many mutations are happening. And it turns out that the coronavirus sequence is changing, but it's not changing very fast compared to other viruses. That's good news for the vaccine front and the antiviral front, because since this virus is sort of has a better copying machine, that means that it's not going to change as fast as another virus might change. There are some changes that you've probably heard about. So there's sort of a famous one in the spike protein where there's a new variant that has emerged that has a change in one of its positions. It's from one amino acid to another amino acid. And there's a recent paper that I saw that suggests that change actually may increase the virus's ability to infect cells. So that's an example of a change that happened and probably emerged because it created a virus that was somewhat more infectious.
Well, we're going to talk more about that later, I believe. And I'm sure we'll get some questions about mutations as well. But so let's quickly go through how our own immune system responds.
Yeah.
To what you've explained, and we'll go from there.
Yep. So we just talked about what the virus is doing, but of course it is doing this in the background of a very sophisticated immune response that has many different components that are geared not only to recognize an infection, but to stop that infection and prevent that infection from happening the next time. And so you can think of the immune response as being broken up into two general categories. There's the component of the immune response, which is operational right away, happens really early after you've been infected with a virus or another pathogen for the first time. This is called the innate immune response. This is a response that is, we would say, not specific. So it's going to behave similarly whether you're infected with a coronavirus or an adenovirus or a rhinovirus that causes the common cold. It's just going to recognize a foreign invader and mount a warning signal. And this warning signal is in the form of chemical messages through molecules that are called interferon molecules. And these are early warning signals that just basically send chemical messages to the immune system that are basically SOS signals. Help, we detected infection. You need to start rallying the armies to come, the cellular armies to come and take care of this infection and stop it in its tracks. And so there will be some cells that are like, that just come as part of the innate immune system. The macrophages and neutrophils, macrophages are called cell-eating cells. Basically, they come and try and engulf and eat infected cells to stop them. But these chemical messages are also activating what we would call the adaptive immune response. And this is a really powerful and specific response that your body mounts against just that virus or pathogen that's in your body. So for a coronavirus, this is a response that says, I'm going to mount a very detailed, specific response to get rid of CoV-2. I'm not going to pay attention to anything else right now. CoV-2 is the laser-like focus that I'm going to develop that response to. This adaptive response takes longer to get rolling than the innate response. They sort of play tag team roles in immunity. One is I'm going to fight early and broadly and send out warning signals. The other is I'm going to develop a really specific, sophisticated response. And this comes in the form of B cells, which are the ones that produce antibodies, and T cells, which a subset of them attack actually infected cells. They're like assassins. So these are the two sets of responses in general that your body is mounting. But you can think of that infection as, it's like microbial warfare that's happening in your body every time you're infected. So this is the body's army, but the virus, every virus fights back. So how does it do this? So let's move on to the next slide. Viruses have to fight back because otherwise they'd be cleared right away. And we know that they evolve relatively quickly compared to us. So they're constantly innovating at the molecular level, not in a directed way. This is all random, but you can select for things that will give them an advantage as they're mutating. So what are some of the strategies that viruses like coronavirus use to fight back? One strategy is, let's just try and hide the valuable parts of our virion from an antibody. So for coronavirus, spike is key, right? That's the thing that if an antibody comes and attaches to spike and prevents it from binding to the ACE2 receptor, that virus is neutralized, it's dead. So the virus is going to try and figure out ways of masking and hiding its spike protein from antibodies. And so it can do this by adding things called glycans or sugar chains, which you can, I think of almost as like a kelp forest. You know, it's a bunch of things flopping around that if an antibody comes in, it might get deflected because it's not there to recognize the glycan. It's there to recognize the spike. And so the glycan might deflect it. So we know viruses like Ebola use these a lot on their surface proteins that are akin to the spike protein to hide them from antibodies. And coronavirus spike probably does this too. And then there are strategies that the virus uses on the inside of its cells, that arsenal of proteins. So for coronavirus, they make about 27-ish proteins from that incoming RNA. And a whole suite of those proteins are directed towards beating down the innate immune system, not allowing the innate immune system to send those warning signals. And coronavirus is exceptionally good at this. And this is one of the reasons why is thought to be linked to its disease pathology, is the innate immune system can't mount, in some cases, an effective SOS response early. But yet then by the time it catches up, it's too late and the immune system is too strong and you get cytokine storm and things like that. So this evasion, this early evasion of the innate immune response is sort of a key feature, we think, of coronavirus pathogenesis. And so the innate immune system, the center panel just sort of describes how that signaling happens. Is a cell will look for evidence that it's got something there that doesn't belong. It's like, what here is not us? What here is foreign? And often it will recognize features of the viral genome that they largely look like molecules that might be in the cell, but have some key differences. And the cell will latch onto those key differences as evidence that they're infected. And they'll use these sensor proteins to detect those differences. Those sensor proteins will then go relay that information to the nucleus to turn on genes like interferon that serve to recruit the other components of the immune system. So the virus actually uses several of its proteins to block those sensors, to prevent those sensors from being turned on, or even if the sensors get activated and the cell tries to turn on those genes that are related to the interferon response, the virus has proteins that will stop those genes from getting made into proteins. It'll degrade the RNA or block protein synthesis of those, just overall to dampen that immune response. And so the virus is constantly fighting against the immune response, trying to keep the cell alive long enough so that it can pump out hundreds or thousands of viral particles.
So there's a whole lot going on.
There's a whole lot going on. It's really molecular warfare that's happening at the, submicroscopic level here between a pathogen and the host.
And everything that we're just looking at right now, for instance, this is happening in every cell where there's some infection going on.
That's
so
right.
There's a lot going on in every cell, and this is happening again and again and again in hundreds, thousands, millions of cells throughout the body.
That's right. That's right.
So
I mean, people who have an active full coronavirus infection, when the levels of their viral RNA are being measured in, per mil, it's in the billions of copies of viral RNA per mil, sometimes a trillion. I mean, really high level of viral RNA that's being produced, and that's being produced in these cells where at the same time as that viral RNA is being produced and the viral particles are being made, there's basically this war going on between the virus and the immune system. And so most of the time we come out on top. That's why we're alive. But sometimes we don't. Sometimes our immune system can't, it gets overwhelmed by the viral response.
Yeah. Well, we're gonna move into how drugs and vaccines may help us with all that. But first, we're going to do our second poll, folks. So here we go again, Macarena. It should be much easier to answer, or maybe not easier to answer, because some people think they have it or had it. I will say for myself, all people are answering, or if you've already answered that, I've had antibody test and an active up the nose test, and it's been negative, so that's good.
I've had a PCR-based test. It was negative at that time. Of course, those tests will only tell you your status, the day you're tested.
Right. That's
you
right.
You can't get a coronavirus test 2 weeks ago and say, but I'm clean. You were
Yeah.
clean 2 weeks ago.
Right.
Doesn't report on your status today unless you got the test today.
So here's our results. And by the way, this is our second poll question, everyone. We'll have one more in a little bit from now. So 86% of you have not had COVID-19. We should make, COVID-19 is the disease, the coronavirus is SARS-CoV-2. Yes, but I'm okay, 5%. Glad to hear that it's only 5% and that 5% has been okay. Yeah, I think I did, but I was never diagnosed. A lot of people now looking back into January, February, March, when they had some really particularly tough time with some sort of infection, are now looking back thinking, yeah, I think I had it, or I may have had it, but of course, as we were just saying, there's no real way to know that. And we're going to talk about antibodies and how long they last in the human body. There was a new paper this morning, actually, saying that they may not last as long as we thought. So in order to be able to understand that, however, let's talk about briefly about drugs, how they can fight the coronavirus, and vaccines, how they will help prevent an infection.
Yeah, so we'll talk about drugs first, and drugs fall into two categories. Category one are antivirals. These are drugs that target the virus directly. The second category of drugs are drugs that are basically immune-based. And so these are drugs that are not targeting the virus, but because we know that a feature of COVID-19, or what really may underlie COVID-19, is an over-exuberant inflammatory response, an over-exuberant immune response, then if we can find ways to dampen that immune response in those COVID patients, then the disease may be mitigated. So there are drugs that will target the immune system. There are actually the two drugs that are being used as widely as possible now, that are sort of have been given the green light by the FDA, of course, would be remdesivir, which we've heard a lot about. That's an antiviral. The way that works is it targets that viral copying machine. It targets the polymerase. It's what's called a nucleoside analog or a chain terminator. It's something that gets inserted into where that copying machine is working, and it jams it. So that copying machine tries to make more copies of the viral RNA, but it can't because it's jammed by remdesivir. And so being able to target that copying machine is a really powerful strategy, and one that virologists and scientists, that copying machine is a popular target for when we think, how do you stop a virus? Well, a great way to stop a virus is to stop its copying machine. There are other drugs that are in development to stop other components of the virus. So there's a protein the virus has called a protease, which is like those molecular scissors we were talking about for activating spike. But this protease actually is used to help activate viral proteins. And so there are many efforts to try and generate inhibitors to that too, none of which are approved and in broad use yet. There are Then for the immune side of things, there's dexamethasone, which is in the class of corticosteroids. These are things that dampen the inflammatory response. So you can think of it in the lower right-hand panel here as a way of, if you have an over-exuberant inflammatory response, the problem is that those cells are coming in and they're starting on that battlefront that's happening in the lung between the virus and the host. there's collateral damage happening to lung cells. And so the immune cells, unfortunately, are contributing to lung damage by damaging sort of neighboring uninfected cells too. So you want to sort of ramp down that immune response by decreasing inflammation. So dexamethasone is thought to do that. There are other classes of drugs so those are the two that are widely used, but many more are in development, of course. So there would be drugs, for example, where, if you could try and find a way to block the interaction of the spike protein with its ACE2 receptor, that could block infection. Or there are many reports now, or reports of many interactions between those coronavirus proteins and many other proteins in the cell they get into. Remember, we have 2,000 different proteins that run the cell, that do the work of the cell, 2,000-ish. The coronavirus has about, 25 or 30 proteins, so a much smaller number, but those proteins are going and basically hijacking other cellular proteins and machines and getting them and pulling them in to do the work of the virus instead of to do the work of the cell. And the virus needs that because it's only got 25 proteins, so it can't do all the work itself. Those proteins have to go and basically commandeer things from the cells. So if you can develop drugs to block some of those key interactions that the virus needs, then you might be able to stall the infection as well. So these are all sort of examples of the many different strategies that are being pursued to develop drugs that might mitigate infection.
Great. We're gonna talk about vaccines too. I just want to remind viewers that after we talk about vaccines, we're going to take one last quick poll and then move to your questions. So if you have a question, please put it in the question box, and we will keep tabs of them. We already are, and we'll get right into that as well.
All right, let's move on to the next slide then. All right, let's talk about this last bit here. Let's talk about vaccines and how do you develop a vaccine and how does a vaccine work? So in order to understand how a vaccine works, you need to understand the concept of what an antigen is. And an antigen may have been a word that you've heard but are not quite sure what it means. Basically, you can think of an antigen is like an identification tag. It's a bit of protein. It's a snippet of, a protein that's part of the virus that serves as an identification tag that tells your immune system about your cells and their potential invaders. And so antigens are really central to the idea of developing immunity because they serve as the identification tags that allow B cells to make antibodies against that specific identification tag, or allows T cells, killer T cells, to attack cells that have that identification tag on their surface. All right, so antigens are really key. So let's move on to the next slide to first describe how this happens. So these antigens, these identification tags, are used as signals. They're used as signals to activate the components of the adaptive immune response. And so these would be B cells and T cells. And so that signal is conveyed actually by a class of T cell called a helper T cell. You can think about these as event coordinators. They're taking that antigen information and they're coordinating the B cell and T cell response. They're sending chemical messages to B cells to produce antibodies and to killer T cells, which are the assassin T cells, to kill infected cells. So the difference between what an antibody does that's made by a B cell and what a T cell does is that antibodies attack the pathogen itself. Antibodies will attack the virus itself, whereas a killer T cell doesn't attack a free-floating virus. A killer T cell attacks a cell that's already been infected with the virus because you want to stop and kill that cell before those thousands of copies of viral RNA that are in there get packaged into new viral particles and amplify that infection like crazy. And so this helper T cell, next component of the animation, will basically send that signal to tell the B cell to produce antibodies against that ID tag. It's kind of an important concept because there are lots of different identification tags. So I often describe this as, think about a car and the surface of the car, right? How do you describe a car? Well, it's got all these different parts. You think of each part as being an antigen. A headlight is an antigen. You know, the ignition is an antigen. A wheel is an antigen. All these parts are being shown to the immune system, and it's trying to make antibodies against all of them in hopes that some of those antibodies will be neutralizing. Some of them will stop the infection, but not all of them will. So not all the antibodies are the same. The antibodies in this example that get made against the headlight, well, that's not going to stop the car, right? It might, you know, flag the car as unusual to other components of the immune system, but it's not going to stop the car. The antibodies that are going to stop the car are the ones that you make to the wheel of the car or to the ignition. And in the virus, those are the antibodies against the spike. And so this is why the spike protein is such a focus for vaccine development, because that's like the wheel of the car. It can't get to the cell or get into the cell without that component. So you need those neutralizing antibodies. All right, so that's the antibody component. The next component of the animation talks about that similar signals are sent to T cells. These killer T cells, which are the assassins that are then going to go and kill cells that have already been infected with the virus. And I think that's going to be shown in this animation here. Here comes the killer T cell, gets the signal that says, go find any cell that's got this ID tag and kill it. And then the final thing is that this is what's happening during this infection, but the body's also thinking ahead. It's thinking, we haven't seen this before, but we might see it again. So we need to remember what we've seen so that we can mobilize much more quickly the next time. We need to have a memory of this. And so for the final animation here, what you'll see is that in addition to having T cells that are and B cells that are components of this immediate infection, there's a population of both B and T cells that serve as memory cells, that their levels will go down over time, but they can rapidly ramp up if they ever see an infection again. So the first time you see a virus, it'll take 15 days or so before these B and T cells are really in full action. So you're really relying on that early innate immune response because it takes a while for this adaptive immune response to kick in. But the next time you see it, either because you've been, you know, your body is getting reinfected or because you've been vaccinated, the adaptive immune response can kick in way earlier, you know, 2 or 3 times earlier or more than what it did that first time, and can secrete many more antibodies or kill more infected cells. So a plasma B cell that's its job is just to produce these antibodies can produce as many as like 1,000 antibodies a second. And you know, there are huge numbers of these cells in the body, so they can be really effective when you hone them to the thing that you want to block. All right, the next slide, let's talk about how do you make, what are the vaccines strategies that people can use to make a vaccine that can elicit this type of response that we're talking about. This is the memory B cell. So the next slide is, so we're going to talk about sort of 2 broad categories of vaccines. The first category of vaccines are those where we as scientists in the lab are generating some form of those antigens, those proteins like spike protein, to show to the body, to show to the immune system. And so this can be either in the form of a weakened virus, where you take a virus and you can introduce mutations or things that make it not able to replicate well, so your body sees it but then can get rid of it quickly. I'll say that this category is not something that, to my knowledge, is currently being explored for coronavirus vaccines, and that's because there are more safety issues with that. The other 2 that are being explored, you can actually inactivate a virus, chemically inactivate it. You know, the genome will be cross-linked and completely useless. There are a number of vaccines that strategies that are using inactivated virus, or you can just say, I'm not going to take the whole virus. we care about making neutralizing antibodies to the spike protein. So let's just grow up lots of spike protein in a big incubator and use that to show it to the immune system and say, here's a protein, make an antibody against that. All right. So in fact, these are under, several of these are under trials now. And then the second strategy, so the last slide here, is that instead of researchers or scientists making forms of those viral antigens, the proteins themselves, you use our own cells to make those proteins. You just provide the instructions for our cells to make those proteins, and then our cells do the work of that and show those proteins to the immune system, those ID tags, those antigens to the immune system to make an immune response. And this is the basis for the nucleic acid vaccines, the DNA vaccines, the RNA vaccine, which you've heard a lot about, that's being spearheaded by Moderna, and Pfizer also has a variant of that, both of these that are in phase 3 clinical trials now, or a safe virus, which is a virus that cannot cause disease in humans and itself can't replicate, but it's basically used to protect a set of instructions for how to make the spike protein that gets delivered to a cell. This is also a vaccine that is under phase 3 clinical trials by AstraZeneca, and the safe virus is a version of adenovirus here. So the difference is that these strategies use your cells to make those ID tags. And the first strategy is you produce those ID tags already in the lab and give them to your cells to see.
Great.
All right.
Great.
I think that covers it in a whirlwind, at least a subset of it. And
that's great, Britt. Really, you're very clear, and your thoughts are organized well. It's really great to to just follow through the tour with you. So, folks, we have one more quick poll, and then we'll get to questions. We have a few questions ready. I see from Jocelyn, from James. We'll get to those. So let's answer this one right here, and then we will see what you have to all say about whether you would accept a vaccine if one were available. And then we'll start with questions. It's also nice to give Britt a little break. We should run music or something, I guess, during these, huh?
I wonder what would be appropriate virus-themed music.
Yeah. Well, see, this is really interesting. I assume everybody can see these results. So just if you can't, or if you can't read them on your phone, if a tested vaccine was made available, would you take it? So 49% said yes right away, 5% said no, and 47% said I would wait for a month to see how others make out. So that's a pretty even split between people who would, go right to the doctor's office or the hospital and say, okay, I'm ready, and just about as many people who would say, I'm not quite ready to go there yet, let me see what happens. I mean, coronavirus is brand new, and the vaccine will be brand new, so that's understandable. Interesting to see that split. I think we're going to get into a little bit about that in a bit. This is our lovely spinning coronavirus, which you'll see while we're doing questions. Britt, something, so I'm going to basically scan questions. I'll try to sum them up. I may name some people who sent them in, which is great. This is from Jocelyn. First names only, folks, that's fine. But it gets to something, Britt, you were just talking about a bit, which is based on the latest news this week about a man from Hong Kong being reinfected with coronavirus. What exactly is happening at the cellular level that allows the coronavirus to bypass built-in immunity and allow reinfection? And that's making some assumptions there in that question. So.
Yeah, that's an excellent question, Jocelyn. Thank you for that. And man, this is a topic that we could spend a whole lot of time delving into, because it's a really interesting one and touches on the unknowns that are really important unknowns right now. I'm familiar with that case, and one of the unknowns is, well, a priori, it's probably not surprising. You could have, even under situations where we know that somebody who gets infected with a virus develops a really robust immune response that largely protects them. In these cases, though, you might not have 100% of the population infected. You might say the majority of the population is protected from reinfection, but there are always some outliers. And so the question here is this individual an outlier and not representative of what's going to happen to most people who have seen the virus? Or the more negative scenario, of course, would be that he's representative of the fact that immunity that is developed after you are infected with the virus wanes rapidly and doesn't protect you against future infection. And this is a question that scientists and physicians really want to know the answer to. The problem is that the only way you can know the answer is to wait and see when reinfection happens. And right now we're just having the first wave of this virus spreading around the globe. And so I think we'll be looking to places like China, which had it first, and may now start to see circulation of the virus returning. What happens to these individuals who tested positive and were sick the first time? Will they, what percentage of them, if any, might get reinfected? And I think that's going to give really important information about how long lasting this immunity is. I will say that many of us are kind of waiting on pins and needles about this. We're nervous because we know that other human coronaviruses, there are 7 known human coronaviruses, 4 of them circulate all the time and cause, somewhere between 15 and 30% of the common cold cases. We know that our immune responses to those circulating coronaviruses don't last a really long time. And that's why people can probably get reinfected with the cold from one of these coronaviruses, every year or two, because your immunity wanes. And so we're really hoping that that is not the case with CoV-2. SARS coronavirus that had the outbreak back in 2003. It's got longer term immunity than the circulating coronaviruses, but again, maybe not super long-term immunity. And so one thing that scientists are doing, of course, are charting. You can measure the levels of antibody and neutralizing antibody in individuals who have the virus, and then hopefully track those over time and seeing what happens to those antibody levels. You're naturally going to expect that the antibody levels will wane some after you've cleared the infection, because you don't need all those B cells to be constantly active. They just need to be maintained at a level where if they see the virus again, they can start producing antibodies. But we don't know what level of decrease of those memory B cells or memory T cells is acceptable to still be able to mount a potent antiviral response the next time you see it, and what isn't. So there's a lot of ongoing research to try and figure that out. And I think without the answers to those questions, we hear about these cases and they make us feel kind of uneasy, but we don't know if they're outliers or representative of what's going to happen to the population at large.
I just want to jump in there. There was a piece out today in the conversation by someone at Tufts University who there's a new paper in New England Journal of Medicine. Sorry if I'm not looking at the camera, I want to get it right here. That's saying that individuals who've recovered, their antibody levels are dropping to half about every 36 days. And that's just one small study. So, but maybe it's representative, maybe it's just the particulars of the study. But I think it gets to this question of what you're saying earlier. In the media, there's a lot of focus on, well, you have antibodies protected, but if they wane, then you're not protected. But they're all forgetting about the T cells and the B cells and the memory. And so I think maybe one of the questions, which has actually come up from another person here, how good is that memory? Is that enough to help you if you get infected again?
Yep, something we desperately want to know, but really can't know because we're still basically at the early stages. I mean, it feels like forever for all of us living through the pandemic. It's been, 7 to 9 months, depending on where you live. It feels like a long time for us, but in the scheme of knowing how long your immunity lasts, and whether you might be reinfected, well, ethically, we cannot reinfect people with coronavirus because we don't have ways of making sure that they would be entirely protected even if they came down with disease. So it's a waiting game then. You can identify people who were infected the first time and then sort of follow them and have them sort of take regular tests to see what happens. But you have to just wait for them to naturally get reinfected and then, or naturally re-exposed to the virus, and then see what their body does in response to that. And I think questions surrounding that are, is your immune response going to be different, better or worse, depending on whether you've had a symptomatic infection, whether you had a severe episode of COVID-19 versus an asymptomatic infection, which we know many people have. And that's on the one hand, it's good that they're not getting disease, but on the other hand, if they don't know they're infected, they can spread to many other people unwittingly.
Right. It's a somewhat related question to antibodies. From James, says, can you comment on convalescent plasma therapy for COVID-19? And this is when we can explain what that is first. And
yes, so convalescent plasma is also something that's been talked a lot about. And what it is basically taking serum, which contains antibodies, hopefully lots of neutralizing antibodies from recovered COVID-19 patients. All right, so if you've recovered, it's likely that you have high levels of antibody in your system, and those antibodies, at least a subset of them, hopefully are going to be neutralizing antibodies. And so the idea is, can we basically harvest and collect those antibodies from recovered patients and use them, passively transfer them to other individuals who are not doing so well with the disease? Basically, can we give them an injection of antibodies that is going to help lower their viral load? These studies are ongoing. There have been, as far as I can tell, mixed results with them. So it's at this point unclear to what extent convalescent plasma, it's not like a miracle cure, that's for sure. There have been some instances where it looks like convalescent plasma may help, and other instances that haven't. And so I think we're just waiting for maybe larger scale studies to give clarity on this. But it's not currently an FDA approved, outside of a setting of clinical trials and things like that, measure, because it hasn't shown obvious sort of unambiguous protective capacity. But right. That's the concept there. Right. And this could be collected from blood banks, for example. I mean, it sounds like something that's like, wow, that's a really inefficient way of collecting antibodies. But through the huge blood bank network, and in theory, I think you could get a lot of convalescent plasma. It's unclear to what extent that's really going to be protected.
Right. This is also related to immunity and what we were talking about just a bit earlier. Constantina, how feasible do you see having a one-shot vaccine that lasts for a lifetime, and therefore maybe eradicating the virus in a few years versus having it maybe like a flu shot where you have to get it every year? And I think the answer is going to be kind of, we're waiting to see that too, but maybe about that.
You're going to hear me saying I don't know a lot, and that's, I don't know because nobody knows. But again, an excellent question. I went and looked and saw what the vaccination regimen is for the vaccines that are currently in phase 3 clinical trials right now. And almost all of those, I think with one exception, are using a 2-dose regimen. And so we'll see what that looks like. And then, the hope, of course, is that you could get one shot and you would have long-lasting durable immunity. And we have examples of vaccines that just elicit a fantastic immune response. actually the smallpox vaccine, which is not in operation anymore because smallpox is actually the only human disease that's been eradicated from the planet. Thankfully, that was a major killer, but that vaccine developed really durable immunity, so you did not have to be vaccinated more than once. There are other very durable vaccines, but we also have lots of examples of vaccines where you need a booster, usually not every year like you do with flu, but you can think of needing your booster for your MMR vaccine, or yeah, certainly flu you have to get every year, things like that. That would be less optimal, but better than nothing. I think everybody would agree we'd take a shot once a year if it, allowed us everything to get back to normal and that protected us against the coronavirus. And so the hope is that at least it will generate durable enough immunity that you won't have to get a booster every year. Maybe every few years you'll need one, and that the virus doesn't change enough so that the vaccine that we develop first, won't be protective against a strain, a variant of the coronavirus that sweeps through 3 years later with a bunch of changes. And all the sampling that's been going on to look at the rate by which this virus is changing and mutating, I think, argues that it's not going to be like flu on this front. It's certainly not going to be like HIV on this front. It mutates and changes at a much lower rate, probably because of that proofreading activity. So I think that, while remains a concern, I think is much less of a concern than how long will the immunity that we get protect us. I think if it's true, I'll say that even if it's true that a natural infection, for whatever reason, doesn't give long-lasting immunity, I think that's entirely possible. That doesn't necessarily mean that a vaccine will fail and not be able to give long-lasting immunity, because it's all kind of in the way that you show these ID tags to the immune system and activate the immune system. And so we don't really know why it is that the circulating coronaviruses, don't generate a durable, long-lasting immunity, but that doesn't mean that we couldn't generate a vaccine that would.
Right.
Excuse me.
Just there's a question or two about flu and influenza and vaccines for flu, and we did a piece online a couple maybe a week or two ago about misconceptions about coronavirus and myths as well. And by the way, everyone, if you want to see this presentation later or see all the things we're posting, just go to scientificamerican.com. But influenza comes up again and again and again. And I believe the vaccine that we have annually protects against 3 different strains of influenza, because there's a lot of different strains. They change quickly. So as you said, you basically have to update that every year and also make a decision. The whole World Health Community has to make a decision about which strains, which 3 they're really going to try to target, because they seem to be the ones that are maybe looking most threatening this year versus past years. And that gets back to the mutations, and you've explained how maybe, hopefully, coronavirus mutates less or less quickly. But there's this terminology issue about strains and mutations and variants. And so you hear some scary numbers about there's thousands of variants of CoV-2, but maybe that's not the right term. Could you maybe just address that briefly?
Yeah.
Mutations, variation, strains.
Yeah, any given virus is going to have a whole bunch of variants that arise, just because every time that genome is copied, there's going to be a random change. But whether or not those changes stay around or drop out of the population because they're deleterious for the virus in some way, that's kind of what it's actually a confusing concept to wrap your head around, because you can think of each virus population as being a group of very, very similar but non-identical entities. They all have small changes in them, and these are not sort of at the level of, genera differences or species differences. These are just variants that are popping up as the virus is replicating. Then there are larger scale differeces where there are whole sections of the virus or segments of the virus that might be quite different, and that's because there's a new variety of the virus that's emerged. So you can think, I said there are 7 human coronaviruses that are in our population now. Well, they're all coronaviruses, but they're grouped into 7 different. There are 7 different viruses that are actually grouped into sort of 4 subgroupings because they have similar sequences, but it's not just 1 or 2 positions that change. It could be tens or hundreds of positions that change that mean they're related but non-identical. So back to the flu example, and yeah, there are a lot of misconceptions about, the relative risk of flu versus coronavirus and, that sort of thing. I think it's quite clear that, first of all, flu mutates much more rapidly. Like coronavirus appears to be, spread at the present, its R0 value, its reproductive number is higher than for influenza, and its death rate is like 10 times higher than flu. Flu will, on an average year in the US, kill 20 to 40,000 people, which is a terribly scary number. So flu can be a very scary disease. I get my flu vaccine religiously every year and most other vaccines that I can. But, coronavirus right now, the death rate is nearly 10 times higher than that. So these are not parallel viruses. And flu is, it's got a different type of genome. It mutates much more rapidly. There are lots of different strains circulating around, not just in humans, but in other animals. And so flu is an unusual problem because you get mixing of viruses in animals like pigs and waterfowl and things like that, which can help make large changes in the virus that mean our immune system won't recognize it at all. And that can lead to pandemics. So we're all nervous about flu pandemics too, unfortunately.
Right. Let me get to a few more questions. There are several different questions about transmission, and feel free, Britt, to say a lot or a little, depending how you feel about this. But can viruses from Hyder, can viruses be transmitted through food and utensils? Xena, is coronavirus sexually transmitted? Those are the 2 specific questions we have about transmission thus far.
Yeah, I mean, you can get virus infection by touching a contaminated surface. So I would say, I tend to be less worried about food, especially if it's heated, because heat will largely inactivate viruses that have these envelopes. So that's somewhat less of a concern, but you can envision that if somebody was infected and they coughed or they sneezed on their hand and touched a utensil or touched a door handle, and you touched it soon thereafter, that could be a route of transmission. I think the major route of transmission is likely to be person to person. So talking in close contact with somebody, especially indoors where, as opposed to outdoors, that's the major risk factor. But it doesn't mean that you couldn't get it from a surface.
Right. And what about sexual transmission?
Sexually transmitted, I'm not aware of any evidence that it is sexually transmitted, except to say that if you're with somebody who's got the virus and they're breathing on you or you're kissing them, then that would be a clear route of transmission. So, yeah.
Okay, great. Weakened immune systems. There's a few questions about that. So let's use Robert's version here. Why are so many people with weakened? Why are there so many people with weakened immune systems? And how does that? Is there a way to tell if your own immune system may be weakened?
Yeah. It's a bit of a complicated question to ask, depending on how I would tackle it. I'd say, let me tackle it by telling you a few things that can cause your immune system not to respond as well potentially. One concern is obesity. unfortunately, and these data I've seen mostly in conjunction with studies for influenza virus, is that obese individuals seem to not be able to mount as effective or robust of an immune response against that virus or vaccine, which is really unfortunate. And we don't fully understand the reasons why that's the case, but that's a really active area of research, obviously, given the propensity of obesity in the United States and around the world, because it means that at least in some cases, these individuals might have compromised immune systems. Poor nutrition can lead to a compromised immune system. And these, I think, are studies that come from poor areas of the United States where individuals do not have good food security or access to fresh foods. And so it's known that if you have long-term poor nutrition where you're not getting fruits and vegetables for long periods of time, your body does not respond to vaccination the same way as an individual who has a healthy immune system. So I think this can touch on immune in differences, can intersect with structural and social inequities that are pervasive across society as well. And then there are, pre-existing conditions that will lead to decreased immune function, various either autoimmune diseases or hypertension or other things like that that can increase susceptibility. So what we would call comorbidities, things where you have already something that is not working well, and maybe your body's energy is directed in part towards that thing and can't expend as much energy towards the new thing. This is a really, just that question in general is a fascinating area and an area that is being really actively researched, but one where we know the correlations, but and much of the time we don't know the actual underlying reason why these individuals might mount less of an immune response.
Right. Okay, great. And that reminds me of something that also came out today. There's new research basically indicating that in the severe cases of COVID-19, it seems like more men than women are in the group that has severe cases. And the insight, and again, I'm sorry, I'm going to read from this study a little bit just so I get it correct, but there's certain kinds of responses. You mentioned the cytokine storm briefly, that in men, that cytokine storm seems to be stronger and therefore is basically challenging the body more strongly than it does in women. And also evidently that when we had talked about memory cells, T cells and B cells, that in women, those are actually more active than men, at least according to this first initial study. So maybe you could talk about the cytokine storm briefly and just mention whether that seems plausible.
Yeah, so to talk about what the cytokine storm is, or how this relates back to what's the link between the activity of your immune system and the actual pathology of disease, the actual how the disease manifests. And the unfortunate fact is that sometimes, actually there's a lot of examples of this for diseases, where the disease is ultimately caused by your own immune system doing too much. So your immune system as a warrior is sort of a double-edged sword. It can attack an invader, but as I said earlier, during that battle, there can be a lot of collateral damage that can injure your own body if it's not brought into check. So it's a really sophisticated and complicated system that is designed to be turned on and then rapidly turned off when it's not needed. And if it doesn't get turned off quickly enough, then it can, cause damage on the body that it's trying to protect. And that's what the cytokine storm is. And that's really thought in part to underlie COVID-19, the serious forms of the disease, is this idea that the early immune response isn't happening well because the virus is doing such a good job of shutting it down and subverting it. And that leads to a delayed immune response, one that's happening maybe later than it should, and an overactive immune response that's maybe compensating or trying to compensate for that weakened early response, but it's an example of too much, too late. Because by the time this really exuberant immune response comes in, well, the virus might be about done replicating and it's moved on to another host. It's still there, but it's gotten out what it needs to get out and is moved elsewhere. And that immune response now is doing less to dampen down an infection, which has already maybe largely been dampened down, and is actually injuring the lung tissue itself. And this is through these cytokines, which can increase inflammation, which is basically a feature of recruiting cells that are going to do this battle. That's what the cytokine storm is. And so the paper you're talking about, Mark, which I haven't read, so I'll have to go and look at it, but just to sort of comment on the concept, which is something that's been discussed a lot, is, it seems like men in many of the countries that are looked at, less pronounced in the United States than in some of the European countries and others for the data that I've seen, but it seems like men are differentially dying and getting severe COVID-19 compared to women. And so the question is, why is that the case? Is it a behavioral feature of men taking more risks, or engaging in behavior like smoking that can is a risk factor for more severe disease? I've heard that argued, but I've also heard argued that it comes down to differences between the male and the female immune system, and that aspects of the female immune system, this is a field of research that's started long before the current coronavirus pandemic, but there are aspects of the female immune system that might be stronger. Maybe those early aspects of the immune system that might be stronger in combating the disease than in men. So sort of leading men to then maybe not have as robust of immune response early, and causing the cytokine storm. But that's speculation on my part. I'm not sure how well, at the detail level, it's understood what the functional consequences of these sex-based differences are.
Okay, great.
And it's also a broad generalization. There are many asymptomatically infected men as well.
Yeah, I have to say, at Scientific American, we're always scanning all journals, and we're on a lot of, we get a lot of notices about new papers all the time. And, people may not realize that there have been literally now thousands of papers written about this SARS-CoV-2 virus. They're just coming out constantly. So, you see one paper that looks really interesting, but science and scientists in general have to take it a little more slowly and say, okay, that's one, there's thousands. We really have to see what's going on across the board before we're going to make a conclusion, so to speak. But I don't know if people are aware of how active all of that is. Right.
It is truly remarkable.
Yeah.
And, and many of these papers are filtering through what are called preprint systems. And this means that people are putting their papers out in the public domain. They haven't been peer reviewed yet, which is what happens normally before you get formally published in a scientific journal, means that your research has been carefully evaluated, usually by about 3 other anonymous but selected by editor scientists who are in the field and can critically look at the paper and say, or the study, and say, here are the changes that you need to make in order to make this scientifically sound before we publish it. So that's what's called peer review. And so once a paper has been peer reviewed, then it sort of has at least a stamp of approval of a set of scientists who have looked at it. But we scientists are always sort of, analyzing them for ourselves. And there's a huge pool of papers that have not yet been peer reviewed. And I think that this is not necessarily a bad thing, right? In a pandemic situation, or normally, oftentimes it's really beneficial to get your information out to as many people who can access it as possible, as early as you can. And that gives people the opportunity to independently evaluate that science and move forward in a pandemic, because it can take 6 months to a year sometimes, once you've written up a study for publication, to trickle through that peer review process. So that is generally not a quick process, and you may want those findings disseminated beforehand. But it is important to note that while these findings are out there, my lab, for example, we put all of our papers on preprint servers so that anybody can read them, but we do it at the same time as we've submitted for a rigorous peer review process. And then those two papers are eventually linked. And so it's, you know, you want to read peer review papers with the eye of, this has not yet been vetted. It doesn't mean it's bad science, but it means that you need to read it really carefully and evaluate what are the strengths and weaknesses of the study, because it has been put out there without having yet undergone peer review. And I think that's really important. And that's a huge component of the massive amount of coronavirus research that's being done now and put out you know, in a timely manner. And so the great thing about, you know, these preprint servers is that everybody can see the data as they're being put out there. And this can really speed up the pace of research and help people build on findings and whatnot. So you have to read them with a careful eye, but it's, you know, I think in general, it's for the best.
I'm glad you got into that, because it's a good window into how science happens. Typically, it's like in this particular situation. I think we can maybe do one more question really briefly, but before I do that, I just want to let everyone know, again, if you want to see this and the whole animated tutorial that was we were using here today, you can see that at scientificamerican.com. Look for us on all of various social media channels where we're posting new information all the time. And those of you who are in this seminar, by the way, you'll get an email with a survey to let us know how we did and what subjects you might like to see covered in future webinars. And there will be a link. This whole webinar will be available as a recording, so there'll be a link in there which you can share to other people, friends on Twitter, or however you might like to do that. I also want to thank Britt Glaunsinger. Thank you so much for being here, for explaining a lot in a short amount of time, and for sharing your expertise today. Folks, you can find out more about Britt online. Her name's right up there on the screen. Just do a search if you like. UC Berkeley and Howard Hughes Medical Institute. And I want to thank everyone who stuck with us today for the webinar. Great questions, by the way. And if you want to investigate further, check us out. And if you want to see what Britt's lab is up to, go to her site at UC Berkeley. We have 3 minutes left. So I have one question that connects your early herpes work with the coronavirus now. And this is something that I think maybe people are starting to wonder about. So certain viruses like herpes can basically hide out in your body for years and then reemerge because something has changed. And it may be too early to know, but is that a possibility for coronavirus?
Yeah, it's a possibility, but I would put it in the unlikely category, I think. You know, the PCR-based testing for coronavirus is quite sensitive. And so it can detect down way below the level of viral RNA that is probably infectious for a person-to-person spread. So while in theory it is possible that a small portion or subset of the viral particles can stick around somewhere in the body and come back, this is not the type of virus that usually does this. So herpes viruses all do it, right? And they do it because they have built-in ways. That's part of their life cycle. That's part of their replication strategy. They have built-in ways to attach their genome to our genomes. They don't integrate like HIV does. That's how it sticks around. But they have ways that allow them to stay and persist. And it's not that RNA viruses can't do that, although it's much more common in the world of DNA viruses. Hepatitis C virus, for example, is a persistent RNA virus. But we don't have evidence, as far as I know yet, that coronaviruses might do that. So I think that's less of a concern, although I wouldn't put it completely out of the realm of possibility.
Right, great. Yeah, no, I may put you on the spot a little bit for that one, but I'm glad. See, there is definitely positive news all through here. A lot of coronavirus reporting is just negative all the time. but there's vaccines being worked on, drugs being worked on. There's more and more evidence being accumulated that we're understanding it better, thanks to people like you, Britt, and your lab, and folks all around the world. So I guess we all need to stay tuned. So thanks again, Britt Glaunsinger, for helping us today. Thanks for everybody behind the scenes running this webinar. I appreciate your help as well. Thank you, folks, for listening and sending questions, and answer the survey, and we'll see you at the next one.
Yeah, thanks for having me. My pleasure.